Hello, dear friends. Welcome back to our coffee break with TRANSFAC. So we'll continue the topic we started last time, which is a very interesting topic, is about model organisms. And yeah, last time we were talking about if you visited our seminar. If not, just you can find us on YouTube. It was about the organism like pig, and we actually were uploading the new genome into our platform. Today we are talking about very interesting organism, which is a Drosophila. It's much smaller, and it's a completely different world. Although the gene regulation, the principles of gene regulation of these organisms are quite similar, and we'll talk about this today. If we look at into TRANSFAC, and we are talking about TRANSFAC today, as you know, from a point of view of how many different transcription factors. The very important part of TRANSFAC are these positional weight matrices, as we call them. These are motifs, which can be used in a special format. We'll talk about it today a little bit, which are going to be used to find transcription factor binding sites in different genomes. And we're collecting in TRANSFAC matrices from various genomes, and of course the biggest collection from vertebrate, for different species of vertebrates, and of course our focus on human, mouse, and rat, and that's the most important model organisms. But of course we're also collecting other motifs for other organisms, and you can see among the biggest ones, like Arabidopsis, the quite number of matrices. Okay, let me go back. Arabidopsis, then like other plants, and also we have quite a number of matrices from fungi. And today we are talking about Drosophila, so fruit fly, and we have a number of matrices for fruit flies as well. Actually, when you analyze your genome, you have to select which area or which kind of not towards the individual species, but actually you have to select the class of organisms you want to analyze, because motifs for the big class, like for vertebrates, or for invertebrates, or for they are grouped together, and they can be applied for various different number of species. All right, so basically, just again, very briefly, how we build these matrices. We are going through various types of literature. We collect individual sites for a particular transcription factor family from a special, class of organisms, maybe from vertebrates or for invertebrates, like in the case today. And then we align those sites and build this, matrix, which is based on how many times you observe a particular nucleotide in particular position. And then we apply those matrices to search in any sequence in genome, in the target genome, to find the potential binding sites. And there are some, statistical measures how to define the cutoffs, at which level to define this as a site or just, not a site. So, and that's these cutoffs has to be defined and optimized. That's all inside our algorithms. And one of the most important algorithms we are going to use today is actually algorithm to find site enrichment. Why do we need a site enrichment? Actually, this is a way to predict which transcription factors might be important for your regulation of your genes. So, for instance, you did your experiment, you have a set of genes which are change the expression, for instance, the upregulated in a particular condition. And then you have actually in the same experiment number of genes, all other genes, or just a special set of genes which didn't change expression. And then your question is, if there's something special in the promoters or bigger regulatory regions of those genes in terms of motif enrichment compared to the background, to those promoters which didn't drive genes to change. And There is a simple, a simple formula how to calculate such an enrichment score and the probability to be enriched by simply counting number of found sites. But important thing here, which is often forgotten, that the cutoffs for those matrices has to be defined. So if you put a very low cutoff, you will get a number of a lot of sites found. If you put a very high cutoffs, then you find very few sites. And which cutoff is actually optimal for the conditions under consideration, we don't know. And that's why we have a special algorithm in this site enrichment program, which automatically defines those cutoffs depending on the set of genes you are analyzing. And the reasoning to do that is that's what something I want to talk today a bit, and that's a principle which is common for all eukaryotic organisms, but maybe also for prokaryotes as well. When the transcription factors, when they're regulating their genes, their target genes, they are not working alone. They're working in a composition of other factors, cofactors, some people call them. And this surrounding, this environment of other factors around the target factor defines the way how it binds to DNA. So, for instance, this AP1 by itself never would bind to this place because this site differs from consensus in 4 positions out of 7. But having another factor like NFAT, which recruit this factor to that place nearby, that's it changes the situation. That's why if we put a cutoff very high for this particular situation, then we will miss this site. That's why the cutoffs are actually specific for a particular cellular situation. And in reality, we have to talk about the bigger complexes when it's not just pairs of factors, but much more. And we have another algorithm which we'll talk today, which look for combination of factors which may regulate my genes together. And this algorithm using machine learning, like genetic algorithms, and it scans the regions of your interest, which can be promoters of your genes, or maybe ChIP-seq regions or autophagy regions of your interest. And that's what we are going to talk today. And it will find out of huge amount of motifs which we are working with, it will find the proper combination. So it uses various functions for that. And the picture you will see today, then when we will have a set of promoters, set of target regions where my factor is binding, and we search for combinations of cofactors around these sites, which discriminate my regions in a so-called composite score from the other regions in genome. And the higher the difference between these two histograms, the better is a model which describes this composite binding of your complexes of transcription factors. So that's like in 10 minutes the work of us for more than 10 years. But that's the essence of what we are doing. We are searching for enrichment of binding motif for transcription factors, and not just single motif, but combinations of them. Okay, so before going into more theory, let me jump directly into the software, and we'll demonstrate it today on a very important, very interesting case, as I said, on Drosophila genome. And for that, let me first go to our website to log in. So this is a geneXplain website. And here you can register yourself if you don't have yet the account in our system, or if you have it already, just sign in. And I'm a super user. I have all these kind of products associated with my account. So depending on your license, you will have the set of products which you can access, including this whole, for instance, if you have just a TRANSFAC database, or you have a full package including TRANSFAC, TRANSPAS, and human PSD. So that's the maximum what we usually offer. And what you always have here is this geneXplain platform. So that's given to you, even you don't have any paid licenses, but just a free account. Still, you are able to access this platform with all functionality in it, all programs. So of course, to add to this platform various databases like TRANSFAC and TRANSPAS, you have to have this license, but otherwise all the functionality is here. So that's how the platform looks like. So that's don't be afraid of maybe too complex interface, but essentially it's very simple. So if you close those tabs, which gives you more hands, more tuning things. But just look at the starting page of our platform. As you can see, this is a very colorful page with tabs which correspond to various type of data you would like to analyze. And what you see here, for instance, you see RNA-seq data, or ChIP-seq data, or just epigenomic data which you want to analyze, or maybe microRNA data, or even metabolomic data, so you measure your metabolites, or you have variants in genome, like you sequence your genome, you sequence genome of your target organism, and you have the whole genome to analyze. So that's you can click on one of these buttons. For instance, if you have RNA-seq data, you click here, and then you get all this list of, like you start from top and going down, of the pipelines, ready pipelines, which will do things with your data. For instance, if you have just FASTQ files and you need to first map them to genome and then do the gene counts and things like that, the read counts, then you have these standard workflows which you can click and run. If you have also things to detect your differentially expressed genes, but the very specific part which we are going to talk today is actually things concerning the analysis of regulatory regions of differentially expressed genes. And there are a number of tools, but actually, in fact, we are going not from RNA-seq data, but that's something let me show you in a moment. We'll start from ChIP-seq data. But generally, let me show you that. Let me show you the data. As an example, today we are going to use this data from Gene Expression Omnibus, which is pretty recent, about Drosophila, experiment. And here, what authors did, they actually did RNA-seq and ChIP-seq together. And in fact, it's a very interesting set of data, and it was published actually in a Nature-driven journal. It's a very high-ranked publication. And the idea of that paper is that actually they were interested in an interesting phenomenon, which is autophagy, and which is we'll touch it a little bit later. But that's a very important phenomenon, which is now very modern. And there was also actually a Nobel Prize given for that mechanism of autophagy. And this autophagy process is important in cancer development. And also, when you are fasting, some autophagy is starting in your cells. So that's very important problem. But it's very difficult to analyze. And it appeared to be that this is basic process, which is in many respects common in human and in many different organisms, including Drosophila. And for instance, there is one transcription factor in human, which is found to be a key transcription factor, which is regulating autophagy. It's actually repressing autophagy. If you inhibit this factor, the autophagy is starting more rapidly, more easily. In that sense, it may be a very good target for cancer on that step, and autophagy is actually pro-cancer rather than anti-cancer. So that's also you have to balance. But what is important is that in Drosophila, there is another transcription factor, which is called M1BP, which is a homologous of that factor. And what they did, they actually removed this one. So they inhibited the endogenous transcription factor in Drosophila, but expressed the human factor inside Drosophila. And imagine there is a, like very foreign protein, which Drosophila genome haven't seen before, never in its evolution. You expressed in the cell, and suddenly these factors start finding their targets in the genes, and moreover, it starts regulating the genes. So that's very fascinating. I would say, I don't know how for you, but for me, I was always wondering how that happens if you just pull some factors, some very stranger, put in this genome, and it's starting actually a very complex function like regulating the genes. So it find its place. And it appeared actually that it started regulating genes, which are in fact regulated by the endogenous factor. Anyway, so what they have here, they've done a lot of ChIP-seq and RNA-seq experiments, but they also provide the preprocessed data, and that's what we are going to do to use today. So, for instance, we will take actually this file, which is differentially regulated genes. When they took this inhibited transgenic Drosophila and compared it with wild type, and detected the differentially expressed genes, calculated log change, and provided this table. So we will see it today. And then we'll also include here the peaks from the ChIP-seq experiment. So basically, they have antibody for this factor, for this human factor, and they did the ChIP-seq experiment and detect, and they also did the ChIP peak calling by themselves, and what they provide as DEG files, which gives you the location of the peaks. As you can see, we are just using already preprocessed data, and it's fine. Although we could also start from FASTQ file directly, go to SRA archive and download those FASTQ files, do the mapping on genome, do the peak calling ourselves. That's also can be done. But once you have such preprocessed data, it's even easier. You upload it and you start from the preprocessed data. So in fact, what you can do, you can simply copy this URL, and when you go to the geneXplain platform, let me go there. Then you can upload your data here. But before doing that, let me open a little bit what you see in the platform. I showed you these pipelines, but on the background in the platform itself, you have all the files located in this tree. So this tree contains all the projects, all of your projects. You see it. I have a lot of projects, but when you start, you maybe have much smaller projects. Number of projects. You can of course create a new project or delete your project. That's what you can do. But once you have a project, then you can put your data here. And just to demonstrate how you put data, first you, for instance, can create your folder, where you want to work, and then you click on that folder, and then you click here to upload your data, and you can put here the FTP link where the data are located on the server somewhere outside. And it's if it's a zip archive, like in this case, it will be automatically unzipping it and then open this, depending on what kind of table, what kind of data you have, it opens it here. So you see it has now this icon, so it recognizes that this table is actually a table of genes with ensemble IDs. In this case, these are Drosophila genes. You see these are fly base IDs, and then you have all the values which were computed by this pre-processing. So this is 758 genes, which are either downregulated, like you see this gene is very much downregulated, and p-value and even a p-adjusted p-value is very significant in 10⁻52. Or there is a number of genes which are upregulated and also quite significant. Here you see also the gene symbol, so to know a bit more what exactly these are the genes. All right, so here we have gene list, and actually the same way you can upload the full BED file with those ChIP-seq fragments, ChIP-seq peaks, which were analyzed in that genome, in that paper, and which I did before. And actually, these are the peaks which I uploaded. And see, it has a different icon now. So this is actually a track. And if I click on that, it opens it in a genome. And it automatically detects what kind of genome it is. Well, you have to define it when you're uploading your file, because the BED format by itself maybe not necessarily contain the information about genome. So when you're uploading the file, you have to give this information to the system. So that's a Drosophila genome. And about the genomes, all the databases which I installed in the platform are here. And as you can see, we have ensemble genomes from various species and various versions of an ensemble here. For instance, human, mouse, rat. But we have also Arabidopsis, fruit fly, zebrafish, Saccharomyces cerevisiae, and nematode. So a few model organisms already, here. And last time, you can watch this, you can see this video recorded last time. I actually showed how to upload a new genome, which is not here. Like in that case, we had a pig genome, and you don't have it already here, prepared in our platform. Nevertheless, you can upload it from scratch. You can, if you have in such case fewer automatic possibilities for the pipelines, but you can do a lot of things with uploaded genome. Here, the genome is already there, so we are pretty safe in using Drosophila, data. So, and what you have here in genome, actually, for instance, you can open it here. So, of course, you have, all kind of tracks. For instance, there is a gene track here, which, gives you the information about the genes. So, for instance, let's jump to one of the nearest gene. Okay, so that's a different genome, right? So that's why it's not here. So the genome I opened actually not the latest one, but this one. So let me put this right. So here we have the genes, and you see these peaks. Let me zoom it a little bit. So here we see the peaks, which are detected by ChIP-seq experiment. So in this region, this transcription factor from human was bound. Well, of course, it's we don't know exactly in which place it was bound, but in this area. So the ChIP-seq is actually in a big area. I have a question, and it's very good that you're asking me. From announcement, I understood you are discussing the proper promoter lengths for analysis today. If there is a proper length, that's a very good question. Actually, I was trying to avoid this question, but you are asking it directly, so I have to answer it. So the proper answer, I don't know. So, but generally, we did quite a bit of computations and found out that the most important regulatory signals, at least in human, mouse, and rat, are located in the very near region near start of transcription. So if you cover something like 1 kb upstream and maybe like 2 or 200 or base pairs, or maybe up to 1 kb downstream from TSS, this area gathers majority of known binding sites, which are really not only binding but also regulating these genes. Saying that, I don't say that there are no enhancers located anywhere. There are plenty of enhancers, but very often that's actually proven that if you have an enhancer for a particular gene, and you know there are particular transcription factors binding to that enhancer, very often the same factor is also binding in the promoter area of that gene. So it's like a, pairing this binding. So, and which means if you focus your attention just on like 1 kb around TSS, then you're on the safe side not to miss too much. And if we talk about model organism, of course, if we talk about like mammalian genomes, the size of the promoters is more or less the same. But if you go down if you go completely to a different class, like a Drosophila, fly, or maybe even plants, we will talk about plants next time, then the promoter size is a good question. So the genome itself getting smaller, but it not necessarily the promoter size is getting equally smaller. So our experience tells us that promoter is approximately the same, and regulatory regions of genes are approximately the same size as in mammalian genomes. So I think that's have something to do with the size of the machinery of the transcription factors and the cofactors and everything which has to be accompanying this regulatory machinery. It has a certain limitation on the size, and that's why you cannot make it too small. All right, so that's just in between answering your question. Okay, anyway, you have here the genes, and you have here the peaks in the regulatory regions of the genes, or maybe even far away from the genes. Like in this case, this peak is located inside the intron of a gene and the 3' end of a gene, but can be also located in between the genes. We, in fact, don't know whether this binding is associated with the regulation of those genes. But assumption, as I told, that if you see a peak which is located near start of transcription, these peaks are kind of a beta peaks. So because again, from our experience, the regulatory potential is peaking around start of transcription. And that's why what we are going to do today is to select those peaks for our further analysis, which are located in the 5' areas of the genes. All right, so how we are going to do that? Let me show. So another question. Okay, that's a good question. What is the advantage of using this platform to see the results of this type of experiment instead of using IGV like apps? Well, that's a very good question. Of course, IGV is a genome browser, right? And it's very, very useful and very powerful genome browser, but it's just a genome browser, that's it. So the platform actually combines tools and visualizations of all kind of biological material, biological processes, starting from what happens on your genome, motifs, genes, motifs, everything what you can map on genome. But then once you map on genome, then you want to understand what these motifs are doing, which transcription factors how they are connected, how they are regulated. So all the signaling and network and pathways upstream of that also can be analyzed with the same platform. So advantage of that platform is that you can combine two worlds which are actually, I must say, almost never combined in any other tools as I know. You either a genomic guy or you are pathway guy. And this platform is actually combining these two. And it's obvious you have to combine because biology is not like genomes is apart and pathways are separate. They are working together. And that's why you have here tools to analyze molecular networks. You have tools to analyze genomic data, and then tools to combine these two. And that's exactly what we are going to do today, to combine gene expression data, which is more or less, expression, just which genes are higher or upper expressed, and the ChIP-seq data, the genomic data, when we know something happening in genome. And that has to be combined. All right. Thank you for your questions. But now let me go on further with this analysis. So as you've seen, we uploaded this table. So we uploaded it here, but I also uploaded it before. And the first thing what you have to do is to select. So this combines 758 genes, which are either up or down regulated. So I would like to analyze up and down separate, because I believe that actually, although you may, this factor, when you express this factor, there are a number of genes which went up and the number of genes which went down. So which means this factor can either be activator or repressor in the same time. Of course, you may say this is maybe not direct, like it's indirect effect of something, and then that's why you see downregulation or upregulation of genes. But in the first place, it's a repressor. It's always a repressor. I would say it's relevant, but if you assume that depending on combination of cofactors, the same factor can be activator or repressor, and that's often the case, I must say, for many different transcription factors, then it stands all in place. So basically, if you have then peaks around upregulated genes, then you would try to understand what are the cofactors which drive the expression of the gene up. And if you analyze the same peaks for around downregulated genes, you will analyze the composition of those sites which drive the gene down, right? Okay, so, and that's very easy. Basically, you have to filter this table, and for that, we switch this tab below. Here we have number of functionalities, like you can filter your table, you can do above threshold, below threshold, depending on the columns you select. Okay, for instance, we want to select upregulated genes, then we say above threshold, and I choose this like log-fold change more than 0, and I can actually add some additional conditions. For instance, I would like to adjusted p-value below 0.1. And then I can write here any JavaScript combination of conditions, and then it is applied to this top, and this way you select a subset of genes, like in this case 366 genes, which are, well, in our case, upregulated. So basically, by clicking on this button, then you save this as a separate subset of your genes. Actually, I did a different. No, in fact, I did exactly the same filtering, and I saved it already before. This is called up. So these 366 genes which are upregulated. But now what I want to find which peaks are located near those genes. And there is a number of tricks you have to do. First, you have to convert this table into location in genome. And for that, actually, there is a function convert table to track. It's open it then in the genome browser when you click it. So now I have these genes. Let me jump in them. these genes, they are upregulated, and you see the allocation in this browser. And in this case, these peaks, which are located in the front of those genes, are very interesting for us. We want to analyze them. Right. So, and to select those peaks, there is one function. You know, I show very detailed things, but of course you can try it by yourself and when you have your account. So I want to filter this track of peaks by this track of peaks of genes to select only those intervals from my track which are intersecting another track. And for that, we have a special function which is called here in data manipulation. We have this filter track by condition or filter one track by another. And then you put here one track, which is a track of your all your genes. Well, all your peaks. These are the track of all your peaks. And then as a condition to filter, I put the track of my genes. So and then you have to specify the distance you want to have the overlap. So actually, if you put very stringent condition, you would require those peaks to be overlapping, only which are located exactly in your genes. But we are interested in those peaks which are going above, which are around the start of transcription and maybe in the introns of those genes. So actually what I did, I specified the region around 5,000 upstream of transcription start sites. And then I filtered those peaks by the genes which are upregulated. And then I got, well, I will not do it right now. I got the peaks which are specifically upregulated. This is a peak which has, this is a track which is specifically located in the, no, this is a track of upregulated genes, actually. Table track, yes, right. And this is a track of downregulated genes. And let me find the track which I'm interested, the track of the peaks, of the ChIP-seq peaks, which are located in the promoters of those genes. Right. So actually, I did it differently. You see, there are many ways of doing the same thing. What actually I did something simple. So I wanted to simply ask, I have my peaks of that transcription factor binding, and I have upregulated and downregulated genes, right? Let's simply compare this, and see how many of them are located near upregulated genes and how many are located near downregulated genes. For that, I have a function which is called Venn analysis. It is actually present here in Venn diagrams. So if you run it, so you can put your set of tables here. And when I did it, I have this kind of diagram which tells me, okay, I have 123,000 peaks in the genome, and I have 366 upregulated and 392 downregulated genes. And I define the region, like the gene itself, 5,000 nucleotide upstream of the gene and 5,000 nucleotides downstream of the gene, so the whole area. And if the peak is located in that area, I grab it. So as you can see, we have here 291 peaks, which are genes. In fact, all right. So this is 123 genes which contain at least one peak nearby. But here we have 291 genes and 321 genes which are upregulated and having those peaks, and these are downregulated and having those peaks. Okay, saying this all very complicated, but as you understand, there is a number of intersection and conversion of those tables and peaks. At the end, I'm now interested in only those upregulated peaks, peaks around upregulated genes. And that's my final result. These are the peaks. So namely 290. Well, in fact, there was one which is somehow missing. I don't know, maybe I have a different condition here. But at the end, I have this 290 peaks. And these are the peaks of the transcription factor binding in the promoters or in the areas, in the regulatory areas of upregulated genes. All right, so we can double click on it, and we have those peaks here. All right, on that genome browser. Good. So what have those peaks? What next? The next is that's something what is very specific what you can do here. So as I said, we want to find enriched motif. Why? Because actually, if the motif is enriched there, it means the transcription factor, if it's active, it preferably would bind to those places. So the enrichment of motif is actually demonstrating, or it's a tool, and kind of a tool to understand which transcription factors are acting in your cell and which transcription factor are actually doing this job, are involved in this case, in autophagy. Okay, so to do that, I simply do the following. I go to the start page, and here in the ChIP-seq section, I go to analyze ChIP-seq peaks, and here we have enriched, identify enriched TFBS in ChIP-seq peaks. And using, of course, TRANSFAC, well, the only pipeline which is offered here. And what I do here, I simply take this track and put it here. I select the species, which so the program knows which collection of the motif it should use, fruit fly, and it immediately selects the proper genome. And here you have to also to select the annotation source from fruit fly. And what is important is, well, in fact, this pipeline doesn't actually know which profile to use, so you have to select it yourself. Although we selected already the genome, but still the profile have to be selected. That's for the cases when you don't want to analyze all collection of the motif from TRANSFAC, but some subset of them. And if you have a special, idea about the cutoffs, what you are going to use. If you want to use the standard thing, what you have to do, you simply go to the TRANSFAC and select here the insect profile. And let me show you this profile. So here in TRANSFAC section of our databases, if you go to profiles, you have this kind of different types of profiles for different organisms. And so we have here insects. So we have, as you can see, 577 positional domain matrices for insects. And here you have cutoffs. And they are defined for two things. First cutoff is a core cutoff. Is actually this is a condition for the very core nucleotides, which are in the center of the motif. And there is an overall cutoff. And the cutoff is from 0 to 1. Cutoff 1 or near 1 means we are interested in very strong sites, so those which are very fitting very much to the consensus. And those which are low, they are not fitting to the consensus, right? Okay, so this is a 577 motif, and we use them all and simply run this pipeline. By the way, you can have a look overview of this pipeline. You can see it here. There are steps like you take your track, which we put here. But as you remember, in our algorithm of finding the enriched motif, we always have to have a background set. So to set of tracks we want to compare with. So if we would have the whole set of genes and we would have non-changed genes, I would choose promoters of non-changed genes as a background track. But since we don't have this data in our hands, we have just already pre-computed list of upregulated genes, then this algorithm is simply create random tracks. So it randomly goes into genome, select any random gene, and pick up a promoter area of that gene as a background track. Why promoters? Because we still want to compare, two regulatory regions, but we want to find very specific features in these particular peaks compared to any random promoter. That's why we choose the random promoter tracks as a background. And then it runs this motif search, it's compared to each other, it's making certain conversions. And by the way, you can also edit this workflow, but that's a separate talk, how to work with workflow, how to add new steps to it, how to modify parameters of that workflow, for instance, analyze the different regions of genome. All right, so then you simply run workflow. I will not do it right now because it takes a bit of time. So let me show you ready results. So you see I actually run it many times with different parameters. Of course, for you, I selected the best parameters to run. And here we see the results. So what actually are the results? The result is, first of all, this list of motifs which are enriched. And it's quite a number of enriched motifs which we found here. So I remember we had 577 or something overall number of motifs, and 178 of them showed a significant enrichment. What it means, which you can see it in this table. For instance, the first motif, which is for the factors STAT92E, so a very narrow family of factors. So you have found 0.2 sites per 1,000 nucleotides of those peaks, and only 0.1, so basically almost 16 times less frequent in the background set. And the p-value of that difference is like significant. It's p in 10 to the minus 4. Not tremendously significant, but still significant. The reason why it's, although the yes to no ratio, the enrichment score is so high, the p-value is not really very high, very sort of significant, because the number of sites actually are very small. So it's only 2 sites per 100 nucleotides, 1,000 actually nucleotides. No, I'm right. Well, right. But if you look at this motif, you see it's quite significant. It's 10 to the minus 13, and the enrichment score is 5, more than 5. And look at this, it's MYBP. So that's exactly the factor which is a homologous of the factor of the human factor which was introduced as a transgene. So actually, we see that in these peaks, which are located near upregulated genes, we have a very significant number of enriched motif from this factor. But not only, it was not actually the top one. So the top ones, as you can see, other motifs which may work together with this motif. And how to analyze the working together thing, which is done by our second algorithm, which I mentioned today, which is this genetic algorithm, which is looking for combination of motifs. Here it is, it's located, here in the same place. So you see, you can identify TF binding sites combination using CMA, Composite Module Analyst. This is using genetic algorithm. There is another tool which is called quite new tool which we created recently, which is called Miller, which is using sparse logistic regression it's a different approach. It's I will not show today the results of this tool, but I'm sure we will have a special lecture about this new tool. It's very powerful. But I will show you results of CMA. So just to show how to start it. So it looks like that. This is an interface to run this CMA tool. So what we do here, we have this track of found sites. So what I showed to you before was the summary, right? So this is summary of all sites found together. But we have also here a file which is called match track, which has actually 2,857 binding sites found in all those peaks we analyzed. And we can actually look at it like as a table. And here we see all the individual sites for various transcription factors, their location in genome, in which chromosome they are, what is the score of those sites, actually, and everything you see there. So you basically take these all sites found and drag and drop it here. As a control, you have to put here the random track sites, right, these ones. And then you specify parameters of that genetic algorithm, like how long it should go. So there are some default parameters. Maybe you don't need even to touch it. The only important thing to consider is actually this thing. So you can run it as it is and can try to find, it will find for you the combination of the best combination of transcription factor binding sites which are located in these peaks. But you can also specify a particular transcription factors in focus. Like in this case, we have this transcription factor, which was M1, yeah, right. This M1BP. So we select this matrix, and we require this genetic algorithm to have this matrix always, well, at least in 20% of your cases of you consider. So which means we are looking for other cofactors which has to be located near this binding site of this factor. And you simply run this, and it will run for maybe a couple of hours. Genetic algorithm is something you have to wait a little bit, and then you get results like that. And that's the final result, which I want to show today. So what you see here. So here are kind of a representation of those peaks which we analyze. Each line here, well, this is one bar, one line, is one peak. It's from here to here is the peak. And this peak is located inside a particular gene, or in front of it, or at the end of the gene. So the genes are showing here as these red bars. And then each individual site is here by a little arrow here. And if you click here, you can see this site, and you can see a consensus of that site. And also you see which combination of sites is found here. And you can see actually those sites are indeed located in a cluster. So that's a requirement which this algorithm has. It searches for such sites which are grouped together, and which means transcription factors which are binding to those sites, they have to copulate. Why you have them in different size? See here, you see very little, right? And here you see them a little bit bigger. Let me increase the font, because simply the size of the peak is different. This is a very big peak. This is a small peak. But the cluster inside the peak is more or less the same size. So finally, out of this 132 enriched motifs, it selected this, how many? 1, 2, 3, 4, 5. These 8 motifs, which work together with this M1BP motif, and they discriminate these enriched these peaks, which are red bars, from randomly pick up regions from a random promoters in genome. So you see, discrimination is not perfect, but it's the AUC is quite significant, and the p-value is very good. So it clearly and significantly discriminate those peaks in terms of by these motifs from the background regions. All right. So by the way, you can now see those sites immediately in your in your genome browser. Simply, where is my genome browser? You can open it here. I already lost. Yeah, it's here. So now you can drag and drop them here. And you see those sites in the genome browser immediately. And well, we can click it there. And, see up to the level of nucleotides to see where exactly those sites are located and what are their score and everything, what you need to understand how this mechanistically regulation of those genes are done through these peaks. Right, so actually, that's where I want to stop today. And especially for genomes like Drosophila, there's not much you can do now from this point. Okay, we know this transcription we found these enriched transcription factors out of the huge amount of them, which can play a role. We found these 8, which most probably are involved in this mechanism. Now that's your turn to do experiments to validate that or to discard this hypothesis. I mentioned in response to one question that this tool can combine also the pathway analysis, and that's something what you can do for genomes like human, mouse, rat, for vertebrates, where a lot of pathway information is collected in our databases, like TransPath. You see here the TransPath, and you see here, for instance, all the pathways which are found in TransPath, and there are a number of algorithms which can analyze these networks and pathways together with transcription factors. So that's the upstream analysis algorithm. For Drosophila, you cannot do much here. So we don't have pathways for Drosophila. So that's why once you get your transcription factors, you may wish to export it to as a set of genes and do some other, you apply some other tools. For instance, you go to Flybase. They provide number of tools to do a bit of functional analysis. Here, as I said, the feature of TRANSFAC is really to focus on motifs and finding transcription factors and their combinations, how they work together. All right, so I have prepared a couple of slides to finish that. Let me see how I can. Right, so I can do it like this. All right, but before, let me answer the question. After today's subject, can you explain what TRANSFAC can do in relation to enhancer sequences, please? Okay. Very good question. So what are the enhancer sequences? That's an important question. So how do you define it? We, of course, in our database, and here, if you go to the TRANSFAC database and look for any gene of your interest, and I put now the human gene because we have much more information about such human genes than about Drosophila genes, but I also have a few information about Drosophila as well. Here, for a gene, you can we have information about enhancers, where they're located, and so we have in special tables collected information about enhancers, which are collected from various databases. And there are a number of approaches how to define enhancers. For instance, in Phantom, they define it in one way. In other databases, they define it in another way. There is a kind of a common understanding what it is. But anyway, we map them into genome. And in fact, at the end, you have a track which contains those enhancers. So in the geneXplain platform, we have a separate track which contains those enhancers. Let me maybe show to you if I have here somewhere open my platform. Yeah. So we have in the tool we have a special project. We have additional information about many important information about various genomes, in this case about human. And we have a track which contained like 326,000 enhancers annotated from various databases. So if you click on that track, you get those enhancers in genome, right? So you have your human genome, and you can put here the genes, and you have your enhancers. Now, these enhancers is just a track, and then it's up to you how to analyze it. For instance, if you have ChIP-seq data, what you can do now in your particular experiment, you can, for instance, intersect these ChIP-seq peaks with known enhancers, and then focus your attention on those areas of those peaks which are overlapping known enhancers. For instance, with this, you may be more sure about that this is not just a simple region where your transcription factor is binding, but maybe it's also playing a particular role because it's overlapping with known enhancer. So we don't have this information for Drosophila, but for human, at least we have, and I believe also for a few other organisms. That's how you can analyze actually enhancers. All right. Okay, I would like to finish simply with, all right, so actually, just, I collected a few slides about this particular case, about this transcription factor. Again, just to emphasize, it's very interesting that transcription factors are so promiscuous. So you put a factor from another organism in this genome, and it starts regulating your genes. And that also means, actually, there was one important thing which I didn't tell you. I did analyze also motifs, or which are known motif for these transcription factors from human genome. And it appeared to be that those motifs are also enriched here in those peaks in Drosophila, but they much less enriched than the endogenous motif from M1BP. Imagine the factor is from human. His pattern is a human pattern, but the motif enrichment, which we see here, are Drosophila motif. So which means in this environment of other factors coming from Drosophila, this human factor becoming more Drosophila, factor. So it's kind of jumping it together and becoming like a Drosophila-like. So the transcription factors are really, really very fuzzy and very, promiscuous in their binding, and that's make them so interesting. All right, so I don't have any further questions here, but I would like to ask you if you have any. So the floor is open for your questions. You see, I opened quite a bit of strings here, and you can touch any of the strings, like what kind of other genomes, or what else do you have? So just ask me, and I will be glad to answer about our tool. Okay, I have question. Are there also curated data sets in TRANSFAC that can be used compared along with own data? Okay, so I don't fully understand your question. What do you mean? What type of data do you mean? So okay, let me show you what type of data we are working. So we are working with RNA-seq data, we are working with ChIP-seq data, we are working with genomic data, microRNA, microarrays even. So what kind of data you mean? What is your data? ChIP-seq data, okay, I see. All right, we don't have, we do have. We do have our ChIP-seq data that's in TRANSFAC database. Which I have already shown to you. For instance, this is one entry from BRCA1 transcription gene, and among, for instance, many things like which kind of disease this gene is bear marking to, and what kind of tissues it's expressed. We have also information about ChIP-seq ChIP-seq data coming from various databases. So it's called in vivo fragments within the promoter of BRCA gene, which are bound by transcription factors, namely 1,795 entries. So it's a big list of those peaks, and actually narrow peaks. Well, what we do, we first map peaks from you see from various resources, like from ENCODE database, from individual papers or individual collections, and then each peak we annotate. We apply motifs for this factor, which is mentioned in that, which is done experiment done for, and we identify exact position of the best motif inside these areas. Of course, that's still prediction, but it's additional annotation. So you have the transcription factor, which was used to do this ChIP-seq experiment, and then the motif from TRANSFAC, which was used to annotate those peaks. So it's a quite a big collection of ChIP-seq data, and this collection is available inside the platform. So what you can do is, well, if you have your own ChIP-seq data, you can compare it to our ChIP-seq data. We also have, it's not the biggest collection in the world. For instance, the ENCODE, well, we're trying to collect all ENCODE information, but there are really dedicated databases in the world which are there to collect ChIP-seq data. We are trying to keep the track, so in order to cover all transcription factors evenly, more or less, but we are not really collecting everything what is there available. For instance, there is another database, a public database, which we are collaborating with. It's called GTRD, Gene Transcription Regulation Database. It seems to be one of the biggest collection in the world, which have the biggest number of ChIP-seq data. And here in the profile, in the platform, well, we don't have a specific collection to compare to. So you go to a database, you can upload your ChIP-seq data of your interest, download, and then you very easily can upload it into this geneXplain platform, and then you compare it with your data. By the way, here what you see, for instance, these are binding sites which are experimentally proven. This is really a golden part of TRANSFAC. I was talking about motifs today, and we of course the biggest database with motifs, but there are other databases which you may know which collect also motifs. But here, what is specific for TRANSFAC, we collect individual factors with individual binding sites from experiments which are done, with a standard and very professional experiments. So you can go to see all details of those experiments. For instance, this particular binding site was done by various type of experiments. We categorize types of experiments. We give a quality to that site, and the best quality is quality 1, which means it's really proven with many types of experiment. It's true binding sites, it's a true regulatory site. So that kind of collection is also very useful. And again, you can access it both from a database interface and from the platform interface. If you predict your binding sites in your genome, then you might be interested to compare whether some of those predicted sites actually coincide with the known sites. And this can be done easily, again, by visualization on genome browser and by filtering them as a tracks. Okay, another question. So the genome used for human is HG38, but is it the canonical version of the whole assembly? Okay. The canonical version or the whole assembly? It's a canonical version, right? So you mean the whole assembly means all the apocryphs, sort of those assemblies which may not fitting into the chromosomes, the canonical chromosomes. Well, it's no problem to upload them. So if you have special genes from those special parts of assembly, you can upload it. And in my previous coffee break, I showed how to upload a new genome or even a new assembly, not ready genome. If you have FASTA file, it can be either uploaded and then you can search. Also, of course, you have to upload a feature table in GFF format, and then you can manipulate and analyze those regions in those genomes. And if you do that, you can upload any FASTA file. So, and in the genome, which we have in the platform and we regularly working, we are using the canonical build. In the meantime, you can, of course, go to our website to see what kind of other products do we have. Some few of them you see here. I already mentioned this TRANSFAC alone, or TRANSFAC as a full package with pathways and human PSD. This is information about this tissue specificity, the diseases, drugs, biomarkers. That's important. But of course, it's for human only. Well, human PSD contains information about other organisms, but the focus is human. Very interesting tool, and we will have a special sessions devoted to that. It's called Genome Enhancer. That's very interesting. So you can read about it in the website. So we try to make a completely automatic pipeline when you shouldn't care about all these intersections, what we will doing today. You have your gene list, you have your peaks, you have to intersect this way, that way, you have to convert tables, you have to do a lot of this routine work. In this Genome Enhancer, it's all done automatically. So what you need is just to drag and drop in one box in the midst of that interface, like FASTQ files for RNA-seq, FASTQ files for ChIP-seq, or preprocessed data, or maybe you have also genomic data, and then define graphically what you want to compare, and then just run it for several hours, and it gives you the full report of the found transcription factors, combinations, upstream analysis, master regulators, drugs, targets, everything in one report. So Genome Enhancer, although it's at the moment just for human, we are working on a version for mouse and rat, but for the moment we have just for humans. So very important and interesting product you may try. Okay, so I think we don't have more questions today. Thank you very much for your attention and very interesting questions. See you next time. Goodbye. Thank you very much.